Okay, hello and welcome to this 23rd lecture on bio-microelectromechanical systems. Today I'll be actually after briefly reviewing what has been done earlier last time, would delve into the process of translation, okay, and then we'll also look into some other aspects related to this beautiful codification process where there would be a change of language from nucleotides into amino acid sequences, okay. We'll also talk about essentially what would be useful for coding what in terms of amino acids, talk a little bit about the degeneracy component in coding, and then compare this whole system of compression of information and translating of information as if it were a floppy drive computer model, and then we will go on to study some other binding mechanisms between different molecules used for immobilization on surfaces, following which we will talk a little bit about enzyme kinetics using Michaelis Menton equations. So let us talk a little bit of the review of previous lectures. So last time we discussed in details about the primary, secondary, tertiary and quaternary structure of proteins, and also looked at how proteins can be formed as enzyme catalyzed processes of condensation reactions essentially. We talked about the various confirmatory stages of structures that proteins have and its importance, and deciphered the importance of the R group and its categories into polar, non-polar, positive and negative charges for essentially the final structure formulation of the protein. We then talked about essentially of the RNA polymerase enzyme and its whole business of how it is used for compression of information from the DNA or the deoxyribonucleic acid and convert that into a compressed mRNA, and we looked into the details of how this transcription can take place. So let me just quickly review the transcription process once more. So if you look at the transcription, really it starts with this RNA polymerase. I call it a magician's hat, okay, the structure here, and essentially there is a sigma factor which comes as an information to find out what would be a region from where the binding and the opening of the DNA chain would start. So it essentially is used to identify the promoter region on the DNA. So this is the DNA, the double stranded DNA, and right here actually, and you have this promoter region here on which the sigma factor goes and bites, okay, and starts opening the chain of the DNA into two. And so the idea is as the chain passes to the other side and goes out of the RNA molecule, it again kind of zips back, okay, it unzips in this region, and then because of the binding of the sigma factor, and then essentially as it binds and opens, there is an assembling of the nucleotides from the solution onto these structures here, and these nucleotides essentially are sutured again by the RNA polymerase enzyme. So what it does is it changes the, or it kind of sutures both the nucleotides, the phosphate groups together by a sugar molecule in between, and then essentially after the 10 nucleotides are kind of operated, the sigma factor automatically loses its grip on the DNA, and then it is of course a process downhill where the unzipping action and the movement of RNA polymerase automatically occurs until it hits upon something which can give a kind of jerk to the RNA polymerase, and it can ask the RNA polymerase to kind of decouple, okay, and so once it decouples by a change in conformation, before that there is an automatic stoppage of the synthesis of the mRNA, which is also kind of defined by something called a stop codon or a terminator sequence. And because of this sudden change in conformation of the RNA polymerase, it also disengages the RNA molecule and sends it out of, like you can see here, the RNA molecule is being continuously generated in this portion, and whenever it hits up a sequence where it can formulate a hairpin loop or something, it changes conformation and detaches itself, okay. So this detaches itself from the RNA polymerase, and the polymerase actually finds out another sigma factor to repeat this particular cycle. But this whole beautiful process is about compressing the information on DNA into this small strand here, which is the ribonucleic acid essentially, okay. So that's pretty much what it is. Let us now look into a very different aspect of this process of translation. Just as the prior process was discussed in the sense of bacterial cells, this process also we will be considering as happens normally in the bacterial cells. But essentially the translation process as kind of applied to the mammalian cells also is similar to this translation process with a few minor variances here and there. But the basic mechanism is almost same as that is in DNA, it's in a bacterial cell. So okay, so how does the translation process take place in a bacterial cell? So you have a this mRNA chain which has gotten out, and from a certain region of as you know, the bacterial cell doesn't have it's a unicellular organism which doesn't have a nucleus essentially. So DNA is present all the way around the inside the membrane, okay, and there are organelles dipped within the cytoplasm, and the cytoplasm essentially also contains all DNA. So all this mRNA synthesis work is inside. There is no nucleus specifically for this purpose. It is just inside the solution, and the mRNA is actually then hits on to the endoplasmic reticulum, where these ribosomes or protein assemblies kind of work on that RNA and try to sequence the amino acids based on the codons which are available. Let us look at it how. So a small subunit essentially this ribosome is one of the most intelligent nanomachineries ever available in nature or given by Mother Nature. It is a small subunit which provides a framework on which tRNA can be accurately matched, and the large subunit catalyzes the formation of the peptide bonds. Okay, so if you look at the basic RNA here, you have I'm sorry, if you look at the basic ribosome here, so this is the ribosome. So we can classify the ribosome as an upper half and lower half, okay, and it is like a jaw. You must have seen these Pac-Man videos where there is a jaw which comes and starts eating the balls or some kind of moving objects. So here you can consider that these ribosomes are like Pac-Mans which kind of move through the solution, and they target the mRNA. And the mRNA is essentially slipped through in the framework on the lower portion of the ribosome and kind of goes there and attaches, okay, and the upper portion is ready to suture the transfer RNA which is there around the solution essentially by matching the sequence of the transfer RNA onto the sequence of the mRNA. However, there is only a set of 3 sequences or 3 base pairs which are available onto one transfer RNA. Okay, so therefore the readouts of assembly of the tRNA on the mRNA is essentially a 3 by 3 by 3 combination. So essentially you have 3 codons at a time, 3 base pairs at a time, which is to be read, and the corresponding tRNA has to be inducted, which also has 3 active sites for binding onto these 3 codons, and then the next 3 groups are taken up and the next 3 groups are taken on, and therefore there is a set of transfer RNAs which are sutured onto this chain. So for the Pac-Man model, the lower portion kind of engages the mRNA, and the upper portion is able to suture the tRNA on the mRNA 3 at a time, okay, 3 base pair at a time. So that is how this kind of protocol works. I am going to illustrate this in details a little more. So let us look at what these are really comprised of. The ribosomes essentially contain about 50 different kind of proteins and several different kind of mRNA structures. It is made up of a jaw-like arrangement with an upper and lower part. Okay, this is how it really looks like. There are so many different proteins folded in different conformations, and also so many different RNAs within this particular network. You can look at it as a set of jaws. This is the upper jaw, okay, Now, this is the lower jaw, and essentially these jaws have 3 different binding sites here, which is represented by, let us say, E, P and A. So, these essentially are pockets. Okay, it is not very clear in the diagram here, but this EPA sites, they are pockets on the upper portion of the RNA polymerase, I am sorry, the ribosome. Okay. So, they are pockets in which essentially as I will see later, the transfer RNA will go and fit really. The lower portion has this rail here, through which you can actually pass a small mRNA molecule. So, the mRNA can go through this rail, and this is essentially the mRNA. Okay. So, you have these different base pairs on the mRNA which can pass through this rail. It goes in here and out of this particular side. So, when not actively synthesized, the 2 subunits of a ribosome are separate and they join together on an mRNA molecule which comes from the nucleus. Okay. And in this case, because it is a bacterial mRNA, it is produced within the solution and it comes to the ribosome. Okay. So, the jaw only closes whenever there is an mRNA. If there is no mRNA, then the rails are open and the jaw is also in the open state. The upper half is open like a mouth. The ribosome contains 4 binding sites, 1 for mRNA, as I already illustrated here, this essentially this area which is like a rail, and 3 for the transfer RNAs. Okay, as E, P and A side, as you can see here in this particular region on the upper flank of the ribosome. Now, very interestingly, there are these bunches of amino acid transfer RNAs which are available in plenty within the cell, okay, within the environment of the cell. So, this is actually a amino acid tRNA. And I will just like to illustrate here that there are about 20 different amino acids which have sequenced some way or the other through this amino acid tRNA. So, essentially this is the protein part of it, okay, amino acid group. Let me just illustrate this through a little more appropriately. So, this part is essentially the amino acid group hooked on to this moiety here, which is a transfer RNA. The beauty about transfer RNA is these 3 groups here for binding. They are essentially 3 among the 4 different groups that normally an RNA would have, that is adenine, cytosine, uracil and guanine. Okay, there is no thymine on the RNA as we told before. So, these 3 groups essentially are bonding sites comprising of 1 or 3 of these molecules, 3 of these 4 molecules. So, based on the sequence here, you can find out a sequence on the mRNA and try to bind this particular amino acid tRNA. But what is important is that this sequence matching essentially leads to development of another sequence here of all these different amino acids, and that is what essentially a protein is about. Okay. So, in step 1, as you can illustrate or as you can see here in this particular figure, the amino acid tRNA carrying the next amino acid is bonded to the A site first. Okay. So, this is the A site here. You can consider this as the A site. Let me just illustrate this a little more clearly. This is the A site. Okay. This particular transfer RNA is essentially with the 3 groups here, and it bonds over 3 different base pairs on the mRNA. Okay. So, incidentally, this 3 base pairs on the mRNA is also known as a codon sequence. It is called a codon. So, therefore, the synthesis of these amino acids one by one on the mRNA is essentially codon by codon. So, you read 3 base pairs at a time and try to match it with the 3 base pairs complementary to the codon sequence present on the transfer RNA, and it has an amino acyl group at the end of the transfer RNA which kind of gets synthesized because of the binding of the tRNA with the mRNA at a particular site of interest. So, this is the site A in which the first transfer RNA is kind of assembled. Now, note here that based on the sequence of 3, the RNA or this ribosome would pick up a molecule, a transfer RNA molecule, which has exactly the complementary 3 base pairs. It will not do something where there is a mismatch. Okay? And so, the very fact that a certain sequence is only determined by the sequence of the mRNA comes very, very clear. So, there is a matching between complementary bases for forming a basis of binding of the tRNA with the mRNA. So, on site A this binds first and then leaves an amino group here. Okay. As you can see in this particular illustration, the amino group is this circle here which has been left. So, let us say you have an amino acid 4 which you are trying to add to this chain. In step 2, the carboxylic end of the polypeptide chain is released from the transfer RNA and at the P side by breaking of a high energy bond between the same. Okay. So, essentially there was a previous transfer RNA which was already immobilized, as you can see here in this particular example, to another that is the P site here. So, this is the P site, this is the A site and this is the E site, as I talked about in my last slide. So, there is already a transfer RNA which has been bonded to this amino acid here 3, which has already formed a bond with the earlier produced amino acid 2. Okay. So, this chain is kind of exiting out of the ribosome in this particular manner. So, you have amino acid 4 here. So, after the suturement of this or filling up of this active site A with the transfer RNA of a particular kind, so as this brings up the particular amino acyl group or the amino group here close by, the polypeptide chain is released from the tRNA, right. So, essentially we are talking about releasing this particular portion of the molecule. Let us just close all this. Okay. So, we are talking about breaking this particular bond here of the molecule. So, it is a polypeptide chain with the tRNA. So, essentially you are disengaging this portion of the molecule by breakage of this high energy bond. This is a very high energy bond between the transfer RNA and the amino acyl group. Okay. And simultaneously what this enzyme does is this ribosome does is that it kind of creates another bond or another peptide linkage between the 3 and the 4. Okay. So, it is not only breaking this high energy bond, but also developing a bond between the 3 and the 4, and this is essentially again a polycondensation reaction. You need an enzyme catalyzed polycondensation reaction here. If you remember from your first slide how a peptide linkage is made between 2 groups of a chain which has OH on the carboxylic end group and NH3 essentially NH2 on the other end. So, you have a H2O coming out. So, it is a condensation reaction in the presence of an enzyme. So, you are making this bond and you are breaking this particular bond, so that you are disengaging the transfer RNA which is there in the site A, the site P, and engaging the amino acid on the bound transfer RNA on the site A. Okay. So, the enzyme that does this activity is also known as the peptidyl transferase. This is an enzyme which would be able to break the high energy polypeptide chain from the transfer RNA and at the same time suture the peptide linkage between the 2 amino acids. So, somehow this reaction because there is a sudden liberation of the amino acid from the transfer RNA and a sudden bonding of this particular amino acid with another amino acid group on the last transfer RNA, there is a huge change in conformation or huge change in size of the particular ribosome. Okay. So, there is a confirmatory change in the ribosome suddenly, due to which there is a change leading to shift of the two tRNAs into the E and P sites respectively. So what happens is that from so this is the E site, this is the P side, and this is the A side, right? So there is a confirmatory change suddenly because of this bond breakage here and this bond formulation here. There is sudden change which leads this upper jaw, as you are seeing, to move 1 unit on the right. So essentially what happens is that if you look at these different pockets C, P and A, the two transfer RNAs shift from pocket P and A to site E and site P. A is left open, okay, and what A does is essentially it takes in another tRNA, amino acyl tRNA from the solution. So therefore the reaction is accompanied by a huge conformation change of the ribosome, which leads to the shift of the two tRNAs from into the E and P sites, okay, and that's step 2 here. So there is a confirmatory change leading to the opening of this A site and closing respectively of the E and P site. The transfer RNAs change their position. Now in the following step there is another conformation change which moves the ribosome 3 nucleotides on the mRNA, okay, and essentially what you are doing is this lower group, if you see here, the lower part of the jaw is kind of shifting in this direction and it changes by 3 groups, and the 3 next groups are inducted inside here, okay. So essentially it is a biting and moving process. So you have this chain of mRNA, so it has bitten, let's say, on these my 3 fingers, and so there are this 3 fingers on the upper jaw of the ribosome, and this lower one has a track like this, right. So you have bitten here, and then there is a sudden conformation state change of this 3, and then following it there is a conformation change state of the lower jaw, and it keeps on crawling like this over the whole mRNA. So there is a continuous rail of this mRNA passing through the lower jaw as there is a slow pickup of the 3 site to the next due to conformation change on the upper half, and then the lower half coming back, then again the next 3 sites for the 3 new transfer RNAs, and the lower half again coming back, and so it keeps on reading on the whole RNA chain like this, okay. So the same process is again repeated after step 3, where a new transfer RNA is inducted, and mind you, all this coding is happening because there is a match between this codon, the 3 new induced groups on the mRNA, and the 3 on the amino acyl tRNA. So what the ribosome really does is a matching of between the transfer RNA 3 groups, 3 end groups, and the mRNA. So that means there is a complementariness which it kind of defines for attaching the right transfer RNA, which would also transport an amino acyl group by amino acyl group, and then these enzymes, peptidyl transferase, etc., they would just cleave off the polypeptide chain and make a peptide between the 2 amino acid sequences. So that way, as this thing is sliding, there is a continuous production of amino acids. As you can see here, this chain grow with time in this particular slide. You can see this chain here growing from this this continuously. You can see that it is keeping on adding these different groups, 1, 2, then this is the 3rd group, this is the 4th group, 5th group, and this chain is kind of getting liberated as the RNA is slowly moving by upper half moving, then lower half following, again upper half moving to the right, and then the lower half again following. So these steps are repeated again and again. So there is a continuous production of these protein chains, and these are of primary interest because the sequence that you are bringing as an mRNA from all the way from the nucleus in a normal cell or in a bacterial cell from the solution in general is really dependent on the portion of the DNA that you are binding. That is number 1. And number 2 is that sequence is translated into another sequence of molecules which are so called proteins of the particular reaction. So this really brings us to a very interesting aspect of cell biology. In a sense, this is probably one of the most fantastic nanomachinery that Mother Nature has provided us. The rate at which the synthesis can be done is tremendous. There are thousands of proteins which are generated in a single second by the several different ribosomes which are floating around in the cell, and at the same time there are a lot of these mRNAs which are being produced on the nucleus, and they are based on the signaling which the sigma factor or some other chemical signals which the RNA polymerase gets, and it gets an information where to start binding and start the production. And so there is a rapid production of all these different groups matching each other, and thousands of mRNAs getting generated every second, which leads to thousands of proteins. And these proteins sometimes get to the surface, express on the surface, and try to get back the physiological state of a cell back into equilibrium. Every time there is some kind of a disequilibrium created by a process, it is essentially a chemical signal which would give you more protein so that the process can be brought back into the equilibrium state of the body. Okay, so looking into a summary of whatever we have done so far, and then trying to do some facts and figures. We know there are about 20 different amino acids that can make an infinite number of proteins. You already know what happens if there is a 25 amino acid sequence. There are about 20 to the power 25 different combinations of proteins which are available. So this is bigger number only for about 25 amino acids, let alone the other proteins which are probably thousands of amino acids knit together. So there the combinatorial is really very high, and you can produce many combinations of these different base pairs together. So 3 bases within the mRNA which can code an amino acid is essentially called a codon. The word codon actually comes from the word coding. Okay, so you are using 3 base pairs successively placed on an mRNA molecule to essentially code a certain amino acid. Okay, and then there are 4 different bases in combination of 3. So if you look at the 3 sites, let's say you have 3 sites here, and you have 4 bases A, C, G and U. So if you put the different combinations that may exist in the sequence, there are 4 that can come on one with another 4 on the other and another 4, assuming that there is a repetition of the particular group. Okay, so there are really about 4 to the power 3 or 64 different such combinations of base pairs which can exist in one codon. So there are about 64 different codons that are possible, okay, based on these arrangements. So 4 different bases in combinations of 3 results in 64 possible codons. Now as we know, there are some terminating sequences or stop codons essentially on the DNA, where on the RNA, where essentially there is a disjointing action because of a conformation change of the RNA polymerase from sliding over the DNA. So it kind of stops the process of mRNA production and disengages the mRNA from the RNA polymerase, and also folds back the DNA or zips back the DNA into a normal double-stranded structure. So these 3 codons are also known as stop codons or terminating sequences. Okay, so let's say there are 3 codons. So how many are really left with? You have about 64 possible combinations, out of which 3 are always used for termination of the process. Okay, so they are termination sequences. So therefore there are about 61 different combinations that may be used for specifying amino acids. But the unfortunate part is that there are only about 20 amino acids, as is explained here in this particular illustration. So there are about 20 different amino acids as you can see in this particular illustration, and there are about close to 61 different combinations of these sequences or codons which are possible, okay, to code these 20 amino acids. So definitely there is going to be some degeneracy. So let us look into some facts and figures. This right here is an illustration of what really happens. So this is the particular amino acid, and this is being coded by these DNA based triplets, okay, CGA, CGG, CGT and CGC, and correspondingly the mRNA codons that are produced out of it are GCU. Mind you, if you look at it a little more closely, there is a complementariness in this. So therefore if there is a C, there is a G on the mRNA, there is a G on the DNA based triplet, so there is a C on the mRNA, there is an adenine here in the DNA based triplet, there is a U or uracil, on mRNA. I mean, again, I would just like to remind you that the uracil is the substitution for thymine group on the RNA, okay, on the RNA molecule. And so therefore, if you see more, there are these CGG on the DNA base triplet corresponding to GCC and so on so forth, okay. So this alanine amino acid is being coated with these many base triplets and these many mRNA codons. So definitely there is going to be a degeneracy. As I told you before, there are about 61 codons which can code only 20 different amino acids. That means there are more than one codons which could be responsibly coding one amino acid, okay. This is essentially called the degeneracy component, okay, of the coding. So there is an arginine here amino acid, and you can see the number of base sequences on the DNA as well as on the RNA, mRNA, which would be used for coding this arginine. And similarly, there is these things go true for asparagine, aspartate, cysteine, glutamate, glutamine, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, so on and so forth. What I am also interested in looking at is these top codons here. If you look at essentially this particular portion, you know, of the molecule, it is actually a stop codon, okay. I'm sorry, let me just do a little bit of, so this is essentially a stop codon, okay. And there are these sequences here which I'll just illustrate here in a little bit. So the base triplets for the DNA ATT, ATG, ACT are essentially the stop codons. This correspond to an RNA sequence U A U A G U G A, okay. So these are essentially the RNA triplets. This is the DNA triplet, and so wherever the RNA polymerase hits one or more of these sequences, there is almost always a detachment, and there is almost always a separation of the RNA and stopping of the process. So that's why it is a termination sequence of stop codons. So these are the stop codons, and so the other 64 combinations which are there essentially are coding these different amino acids, these 20 different amino acids. So it is really like a mini computer system. So I would like to draw a very strong analogy between what happens on a computer as to what happens on this DNA as you do the transcription and translation. So on a computer, if you look at, you know, you have something like a floppy disk, okay, and you can consider that in this DNA codification and translation process, the chromosomes are like floppy disks. They are essentially readouts of data points where this compressed DNA, this compressed all this information is present, okay. So on a floppy disk you have files, you have several files to store, whereas in the chromosomes you have these different genes or these different DNA which are supercoiled and placed inside the chromosome. So chromosome is kind of equivalent to the floppy disk here, and the gene is kind of equivalent to the file on the floppy disk here, okay. And so therefore if you consider a file, really it is made up of this electronic information called bytes, okay, and one byte is basically an 8-bit character. A bit could be either a 0 or a 1, right. So you have like an 8-bit character which comprises a byte, and then there are several kilobytes, megabytes, you know, those kind of things which can be the size or the total information on a single file, as you can see here, right. So the bit here is essentially either a 0 or a 1. Correspondingly, if you look into a gene, really it is the codon of these 3 base pairs on the mRNA which is comprising of the same information as is there on a byte which can correlate to a file. In this case, the codon is the information, a 3 base pair on the DNA or a base triplet on the DNA which is a codon, which actually comprises of the gene. Several of these codons altogether comprise of the gene, just as several of these bytes, kilobytes or megabytes, comprise of the file here, okay. And just as in a byte you have about 0 or 1, 2 different characters, 8-bit character, where the characters could be, or 1 bit could be either a 0 or a 1. So you have 8 such numbers or 8 such combinations of zeros and ones for representing 1 byte. In this case there are these 4 different combinations, A, T, C and G, adenine, thymine, cytosine, guanine, which are the 4 base pairs on DNA which comprises of this codon. So these 3 bases are essentially made up of 4 rather than 2. So do you see the complication here? In a computer, what is done with 2 characters, in real life in DNA it is done with 4 different characters, and the computation essentially just increases 2 to the power 2 essentially, okay, times. So you are considering here instead of zeros and ones 4 different characters to write a bit of information, and then from that 8 bytes, 8 bits are taken together to a byte, and here probably several of these bases are taken together to make a codon which is 3 base pair long, okay. And so what is important here for me to tell you is that a corrupted file in this kind of a case may be something which has an information or a link missing, okay. In case of a DNA, this corrupted file is a mutation where there is a certain sequence mismatch or there is a certain sequence redefining which may modify altogether the way that a DNA performs in all its processes, including codification, including everything that it particularly does. So there is definitely a correlation between the DNA, or between what the DNA does and what a computer does, okay. And so this is really very interesting model which shows how the human machinery can behave in a pre-programmed manner, and in fact the way that a cell dies also is pre-programmed or prior programmed inside the cell using this information, and that is the most amazing part of what we are really, okay, and what probably Mother Nature has made us of, so that we have predefined timelines of existence. So some philosophical thoughts on that, and then let us look into another kind of summary of the whole transcription translation process. So in summary, what we would say here is that essentially the hereditary information is encoded in the chemical language of DNA, okay, and this is also a basis of reproduction in cells and all living organisms. The DNA essentially, as we know, is comprised of 4 basic groups: adenine, guanine, cytosine, thymine, okay. And in all living cells essentially this double stranded DNA undergoes the process of transcription and to form single stranded messenger RNA, and it is a much more compressed code of information than that on the DNA itself. The mRNA of course is comprised of adenine, guanine, cytosine and uracil. Uracil is the new group which is added on to essentially all RNAs, and which kind of replaces the thymine on the DNA, okay. So that is how they are built, and then the mRNAs which are generated this way undergoes this translation mechanism in the ribosome, wherein a group of amino acids are stitched on the basis of 3 base pairs at a time, or a codon in the mRNA, and that way you can have a sequence of amino acid from a sequence of mRNA essentially. So that in a nutshell what all this process is comprising of. You can look at the nice cartoon here on the right which talks about this DNA structure. As you can see, with the help of a RNA polymerase you can generate this RNA, and from the RNA again you can produce a protein using ribosome as the enzyme. So some facts and figures or some take homes are that DNA essentially comprise of ACGT RNA, ribonucleic acids comprise of ACGU, nucleotides are bases essentially in ACGTU, all of them. Proteins are made up of 20 different amino acids in different combinations, in a variety of combinations. The RNA polymerase synthesizes the mRNA from the DNA, and the ribosome synthesizes the protein from the mRNA, okay. So these are in general take homes. In summary, I would like to say the whole area that we have been discussing so far of this translation and transcription is essentially based on or revolving around the fact that the nucleoside sequence of DNA and its expressions in various cells is of utmost importance, because every diseased state essentially or biological function of a cell could be traced back to a single or a group of genes, okay, as DNA sequences. So it is very important to find out how a cell can be able to revive itself from a disease state into an otherwise equilibrating stage where they do not perform any malfunction, and this whole route of conversion from a DNA to an RNA to a protein is essentially helping it to do that. So, the determination of the signaling pathway of proteins is very vital to the understanding the function of cell. This is essentially what the signaling pathway would mean. Okay. So, information in DNA is pretty static, and it does not change much with time except when there are mutations. However, the mRNA which is transcribed onto the DNA is a continuously changing, continuously generating form of molecule. So, several copies of this would be produced time and again, and the translation processes are very dynamic along with the transcription processes. So, it is not that stable as the DNA is. It is a very metastable molecule, and therefore sometimes in detection assays also it is a better idea to look at the messenger RNA instead of the DNA as a basis of detection of the particular entity, biological entity. Of course, these two fields of trying to study what is on the gene and trying to code a particular protein, trying to study what is on the amino acid in the protein, they do make up this area of genomics and proteomics, and these two standalone have very wide applications as on date in the field of biotechnology, medicine, agriculture, biology, etc. Okay. So, therefore, in a nutshell, this is what the life process all revolves around for kind of getting back into an otherwise healthy state from the disease state. So, if you could work out in a better way on this whole this principle of transcription and translation, and could identify the individual pathways which would cause these signaling to happen to produce proteins or not to produce proteins or produce at a certain rate, you could control diseases essentially within the human body. And therefore, this whole area of systems biology now is actually more concerned with working at biology from a systems point of view, wherein all these information pieces that you get are kind of stored up in an organized basis where you could have a proper control on the signaling processes, on the way that the RNA would be essentially produced from the DNA, on the way that the protein would be produced from the RNA, and any of these mechanisms, if disbalanced or cut short or changed or altered, can produce altogether different set of processes for the cell. So, looking from that angle, the system biologist kind of derives his theory and database accordingly. So, we are kind of towards the end of this transcription translation process. Another very interesting factor which I would like to discuss before moving on to the next topic is that how proteins can be used as linker molecules, okay, and how they can be used to immobilize different moieties, biological entities onto the surface. And for that we need to study the protein complexes as biolinks. Okay. And so, we have just a few pieces of information put together here which talks about, and this paper, this essentially has been taken up from variety of literature which is available worldwide. There are certain references which are also quoted here, and essentially this paper on adsorption of avidin on the microfabricated surface for protein biochip applications has been used for citing this particular slide. So, there are several biolinks that one uses for doing or capturing cells or different biological entities on a surface. Why is capture needed? Of course, because we have been talking about the whole area of electrochemistry, of sensing. Essentially, you have to immobilize something in order to find out more details about the information contained in that thing. Okay. So, immobilization is a primary goal of any biosensor. How you can immobilize a recognition element, how you can immobilize a particular biological entity. Okay, that is the first primary goal of a biosensor. So, the first such biolink of course is this chemistry between antigens and antibodies. Okay. So, as we already know that antibodies are essentially proteins which has a complicated Y shape. So there are two light chain sides of the chain, which are essentially arms of the Y, and then there is a heavier chain which is at the bottom or stem of the Y. Okay, and this kind of linkage on the lighter chains, you have these epitope sites or chemical sites which would be able to recognize sometimes selectively in case of monoclonals, and sometimes non-selectively as in case of polyclonals, the surface proteins of these antigens. So antigens could be some infecting pathogen cells, etc. So here, as you look at this balloon like feature here is essentially the antigen, right? And you have these two moieties which are the lighter chain moieties, and this is the heavier the chain of the Y, stem of the Y. And right along this particular region here on the top, you have so called the epitope sites. Okay, so let me just quickly change the ink color here so that it causes better visibility. So this is essentially what is there on an epitope site. Okay, and similarly you have another site here in this region which is also an epitope site. So the way it works is that the proteins which are on the surface of this particular antigen here would be able to bind specifically to these epitope sites. So therefore, whenever there is an antibody which is somehow immobilized somewhere, you could bind two or more of those cells on the basis of their surface proteins using the epitopes which are there on the antibodies. So the binding could be through hydrogen bonds. Okay, it could be through ionic or van der Waals or even covalent at times among the proteins on the surface and the epitope sites. Okay, so that is one mechanism where you could actually use this as a molecular hook for trapping flow samples. Now how you put the immobilize the antibody on a surface is another kind of backwardly integrated process which I am just going to talk in the next few minutes. And then we have this ligand receptor binding. Okay, what this essentially means is, and this is one of the very famous locks in biochemistry, it is called the avidin biotin lock. So you can see how the structure of one subunit of avidin really looks like. It's a very complicated essentially molecule, as you can see here, and biotin is essentially again an organic molecule. It's a vitamin. Okay, so you have a bonding between the avidin and biotin where the affinity constants are as high as about 10 to the power 15 per molar. So they are commonly used in assays as molecular locks. This is something that biochemistry has to offer. Also very important is the binding of some kind of an antibody using this concept of biotin avidin chemistry. So here what you do is, let's say you have a surface which you want to bind something on, and you create a protein in its positive state at a certain pH. Okay, and so once the protein is protonated, you can actually create this charged surface of the oxide to have the exact opposite charge, so that there is a binding of the protein. Okay, in this case the protein is BSA, bovine serum albumin, onto the surface of the oxide, as you can see here. All right, and what you also do is that this BSA essentially is somehow biotinylated. So you have a biotinylation done over the BSA. You put a biotin moiety on the BSA, and on the other hand, let's say this is some kind of a biotinylated molecule, maybe it is an antibody. You biotinylate intentionally the end group here. Okay, so you have a biotin on both ends, one in the BSA end, another in the molecule that you are capturing in that end, and you are using an avidin linker between these two biotin groups, one on the BSA and one on the antibody, and you are actually trapping the antibody in this manner onto this surface. So whatever you have done here, which is concerned with probably the upper portion of this molecule in trapping the cells, the lower portion is fixed using the biotin avidin chemistry, using some kind of a species which can adsorb or physisorb or ionically bond, electrostatically bond itself to the surface, the oppositely charged surface, any surface in fact, silicon dioxide, any metal surface, so on and so forth. So, this is a very interesting approach of how you could actually bind protein complexes to a surface or immobilize protein complexes to a surface and further immobilize the different biological entities by using links like antibodies to identify or select. So, common mechanisms like ELISA we have looked at before also consider this antibody antigen kinetics in order to determine a change in coloration from where we can figure out what is the immune response of the patient or whether he has been afflicted by HIV or not. So, now after doing this we would like to do a little bit of more work in understanding how enzymes behave, and for that we need to find out some mathematical formulations related to the rate constant of various reactions as the enzymes catalyze these reactions. As I have discussed many times earlier, the enzymes by themselves do not react permanently to substrates, but they do participate in the reaction and create an intermediate complex with themselves as a part of that complex, and then later on they dissociate the molecule making a product and dissociate themselves completely as a free enzyme which are available for the next cycle. Okay. So, really if you see at definitions or look at definitions, the enzyme is a large complex macromolecule consisting of largely of proteins and one kind of prosthetic group which could be either a non-protein or a non-amino acid or even sometimes organic or inorganic group, maybe metals sometimes. So, the other part by and large is made up of proteins except one which is a non-protein, non-amino acid kind of moiety. That is what an enzyme essentially is. So, if we look at the basic enzyme catalysis mechanism, let us suppose we start with the substrate here, and by substrate what I mean again is some molecule which we need to modify into a product, and you have an enzyme E, and there is a binding kinetics going on between S and E at a certain rate K1, and this is in equilibration with a intermediate enzyme substrate formulation which has been formulated here, and then there is a equilibrium setup. So, there is a reverse rate constant which is also same as the forward rate constant, and there is an equilibrium between the enzyme and substrate independently and the enzyme substrate complex. Now, if you look at what happens to this complex is essentially again converted into an enzyme and a product, okay, and there is a rate constant K2 in the forward direction in this case. We are not concerned with K minus 2 here for reasons which I will disclose a little bit later. So, we have from substrate to enzyme and enzyme to product. The enzyme essentially is retained as it is and the substrate is converted into the product or catalyzed by this enzyme. Okay. So, that is how this whole enzymatic complex formation process is defined. Now, let us look into some examples, like for example, we have been talking about this enzyme GOD, glucose oxidase. So, when you convert this or when you mix this with glucose and oxygen and you break it down, there is an enzyme substrate complex which would be formulated and which would generate the back into the GOD, the gluconic acid and the H2O2, which are products of this particular reaction. So, the enzyme gets separated as it is. Now, we want to apply the steady state approximation of this reaction system as borrowed from the kind of kinetic theory. Okay. So, we want to find out what in terms of concentrations of various substrates, enzymes or enzyme substrate complex can we really model the rate constant as. Okay. So, some approximations, we assume that during most of the time of the reaction the concentration of the complex, the enzyme substrate complex is very, very steady, that is constant essentially. So, that is one approximation that we make here, and then also from this we can derive the fact that the rate of formation of the complex from its components is balanced by the rate of breakdown. So, there is an equilibrium between formulation of enzyme substrate and formulation of the enzyme product. Okay. and also there is an equilibration between the formulation of enzyme substrate and the kind of back formation of the enzyme and substrate individually. So there is an equilibrium in the forward and reverse direction as you are forming the complex, and then there is equilibrium in the forward direction as you are forming the enzyme product from the enzyme substrate. So the rate of formation of the complex from its components is balanced by the rate of breakdown back to enzyme forward to its product. Thus we have the rate of formation of the complex as kind of represented in this kind of a manner. So K1 is the equilibrium constant, so you can have the rate of formation of the complex, that means dES by dt. It's a positive rate, so you are formulating essentially this K1, which is the rate constant times of the concentration of the substrate times concentration of the enzyme minus the reverse rate, which is K minus 1 times of concentration of ES, which also shows the breakdown of ES into individual enzyme and substrates. Okay. So on the other hand, the rate of breakdown of the complex essentially into the enzyme and product. Okay. So this is called the breakdown of the complex into the enzyme itself and the product. That is dependent on the forward rate at which ES complex is converted into the enzyme and the product. Okay, so that is K2 times of ES. This is K1 SE minus K minus 1 ES. All right. So let us look at some of these formulations here. Let me just write it down once again. So we have K1, K minus 1 and K2 as the forward reaction rate constants. Okay, so I think because we are just more or less towards the end of this lecture, what I am going to do is to pick it up this particular topic in the next lecture and try to derive what you call the Michaelis Menton equation. So I would like to close the lecture with this. Thank you.